Hey, good afternoon. Um, Sorry, a little late on the videos today. Going to be going through unit, I think again, with unit five. We're looking at hereditary information, heredity, which this, along with the next unit, kind of genetics unit, I would expect there to be quite a bit of, I guess, information on. But again, with the two FRQ questions, we're not really certain as far as what it's going to be. But so we're going to split this up. First one, we're going to talk meiosis. Second one, we'll probably go through the Mendel versus non-Mendelian genetics. And then third one, we're going to talk just a little bit about some of the problems, the laws of probabilities, and Punnett squares and pedigrees that you may need. But again, just kind of a refresher course for what maybe you guys will need to know, or what you guys can go out and study a little bit more. So first and foremost, we talked mitosis a couple videos back, and how when we talk mitosis, we're actually building up cells, somatic cells that we find inside of the body. Remember, in meiosis, what we're looking at is we're looking at the production of sex cells. So whether we're talking sperm, we're talking eggs, they're going to go through virtually the same process here in with some variations in building up those cells. But I think the biggest thing you guys need to understand is the differences between mitosis and meiosis. So again, meiosis two division. There's actually reduction division in that first in meiosis one. Virtually they're the same process in that you've got PMAT occurring. Just again, the overall purpose of meiosis is that we want to reduce the amount of chromosomes in half. We want to go from 46 to 23 if we're talking respectively about humans. We want to go from a diploid cell to a haploid cell. And so again, as we go through, think about those purposes, reasons why. What is the reason why we want meiosis to occur? And again, a lot of it is due to genetic variation. We don't want a lot of variety within our cells that make up our body, nor are they going to when virtually they start out as one cell in the zygote, fertilized egg, and then eventually work into what they are. But if we start looking at meiosis, we want genetic variation. So along with sexual reproduction, we've got a couple things that happen in meiosis one that will lead to that process. One being independent assortment. We've got crossing over that takes place, and then we just throw in the process of random fertilization of just the amount of individuals that we have in the world reproducing with each other, and just what are those possible combinations that could come together. So again, genetic variation is what it's coming down to. So for you guys, I think when we go through and talk meiosis, the big part is going to be again just that situation of what is occurring. Meiosis one. Okay, just briefly go through that again. PMATs can occur, but a little bit different in the case that it is reduction division. You've got the sister chromatids will come together, and they're going to form homologous pairs, which basically is going to be this tetrad that we're seeing right over here. Okay, so you've got sister chromatids that'll pair up. They're going to basically join together at these places called chiasmas, and therefore they form this homologous pair and this tetrad that we see sitting right over here. So chromosome one from the male, chromosome one from the female are going to line up, 2, 3, 4. And remember how they line up down the center of the cell and pair up. It's all random. So male could be on this side, female on this side. We talk about chromosome 2, it could be vice versa, and so on down the line. But again, just really leading to that whole, kind of random, assortment of these individuals, independent assortment that they call it, leading to that sexual, or essentially sexual reproduction, but leading to what's the word? Genetic variation. Okay. So all in meiosis one, we're looking at reduction division, going from a diploid number to a haploid number. And then we're also looking at basically, in this case, the homologous chromosomes separate. And so again, how they, we're looking at right here is that eventually you've got the homologous chromosomes separate, and you're going to find, male go to one side, female goes to the other side, or vice versa as it goes through. The meiosis two is just all about the division of the sister chromatids. Now, when you go through and talk about kind of meiosis one, meiosis two, in terms of oogenesis or spermatogenesis again is going to perform 4 equal cells. Oogenesis is going to kind of produce a so-called super egg with 3 little polar bodies that float around beside of it. So a lot of combinations. Shouldn't say a lot of combinations, but a little bit different in how in what we see between the male and the female reproductive or gamete, aka sex cell. Probably the biggest thing again that we start looking at is crossing over again happening in that meiosis one, just remembering what occurs basically at the crossing over. We have one arm from one of the chromosomes is going to exchange, or it's going to basically take the place of the arm on the other non-sister chromatid. Again, going to lead to genetic variety. Just a lot of, again, possibilities that we start looking at with meiosis. So again, as you guys go through differences between mitosis and meiosis, obviously you start looking at the cells that are being produced. We start looking at a genetic variation. There's a lot of similarities in the PMAT, but there's a lot of differences as we go through as far as the two divisions and what's really occurring. In that the first step is all about the separation of the homologous chromosomes, and then we start looking at the second division being the separation of the sister chromatids themselves. So again, any questions on meiosis, feel free to let me know. And we will move into our Mendel versus non-Mendelian genetics here on the next video.